Hello. But you can see our to-do list on the board. As always, when we tackle a new system, we'll start off with the functions of that particular system. Here you go. And then,
oh, I have this one.
Got one already?
Yeah,
all right.
Thank you.
No problem. So we're going to list the functions of the urinary system, and then we get into an overview of the anatomy. I've kind of started a diagram over there that's going to help us map out the blood flow through the kidney. If you look through your class handout, the one that's up here, I think it's the page number 2, the back side of that first page, you've got a flow chart there of the blood flow through the kidney. So you can kind of list the parts that I'm going to try and draw on the board on that page too. And then you'll notice even after that, you've got some diagrams on there that will walk you through the nephron. We're going to call that the functional unit of the kidney. In fact, all of these functions that we're about to list that are functions of the urinary system, those functions are carried out by that structure, the nephron. So once we kind of build up blood flow through the kidney, kind of like we did blood flow through the heart, we'll talk about where you could find those nephrons, and then we're going to sketch a nephron, kind of like we sketched that GI tract, that 9-meter tube, and we split it into different sections, and we had to know the sections of the GI tract and what was going on in each segment of the GI tract. We're going to do the same thing, but with nephrons, and we'll talk about what's going on in each segment of the nephron. We'll see some of those in the lab, so like I mentioned, the lab list is out there. And quiz 8, which is all about the stuff that we talk about today, is currently on D2L. That reminds me that quiz 7 about digestive system is still up. I think it goes away on the 7th at midnight, so be sure to take it if you haven't done so already. This one I think has been available too, so hopefully you've had a chance to take that. But we just now are getting into the information, so stop me if there's any questions about any of this stuff or any stuff from last time. I'm reminded probably the main thing that you're thinking that I don't have on the board here is the return of exam 3 and going through the answers. And I do have those to pass back. And I did just quickly highlight the answers on one of the versions so that we can flip through those. But since my brain is already kind of going into urinary system, I was hoping that we could get through that first in the lecture part. And then after we run through the lab models, we can kind of switch our brain back to exam 3 stuff. Is that okay with everybody? All right, I'll take that as a resounding yes, and we'll launch into this stuff. Let me see if this thing is working. Yes, all right. Then I'll just try and park myself over here so I'm out of the way, and we can talk through some of these slides. The first ones take us through the functions of the urinary system, and we can really think of these as all functions of the nephron. We're going to zoom into the different parts of the nephron to see where each of these things are occurring in the nephron, but we're going to see overall the urinary system is going to have to regulate water balance. We mentioned last time, and might as well put it on the board again, that there's this direct relationship between blood volume and blood pressure. So by maintaining blood volume, one of the things the urinary system is also going to help to do is maintain blood pressure. Okay, so we've got those things on there, blood volume and blood pressure, the last two things. That ties into regulating water balance. By regulating how much water you have in the blood plasma, that regulates blood volume, which is tied into blood pressure. So we kind of crossed those 3 off the list. In addition to regulating water, we're going to see that some of the cells at the distal end of those nephrons can actually regulate what ions are kept. Instead of ions being wasted in the urine, there are cells right before they get out of the nephron that can reclaim certain ions. They can reclaim things like hydrogen ions if the blood is too alkaline. It can retain potassium ions or sodium ions. So we're going to see what cells are responsible for this ion regulation. The cells that are able to hold on to extra hydrogen ions or get rid of hydrogen ions are going to help regulate blood pH. So remember that pH just had to do with how many of those free hydrogen ions there are floating around in the plasma. So by regulating this particular ion, it's going to help regulate the pH of the blood. Almost. I'm going to have to let that one go. Okay, so then we see how pH and ionic regulation kind of go hand in hand. The other thing we're going to see is the elimination of metabolic waste. primarily things like urea. That's a byproduct of protein digestion. Proteins are long chains of, there's 2 carbons followed by nitrogen, and then 2 carbons followed by another nitrogen. So if protein is your main source of nutrition, then you have a lot of nitrogenous waste that has to be disposed of. Urea is how that is gotten rid of, and it's eliminated, we're going to see in the nephrons. So I think that is all we need to mention on this slide, but now I have in my head, I'm reminded of some of the things. Maybe we can go ahead and put that on the board. We've got a hydrogen ion right here. And we know what we're going to do, so I'm just going to go ahead and get rid of this if that's all right with everybody. Just talking about nephron stuff. We'll go through the exams as soon as I get done with our introduction to nephron. What I'm wanting to put up right here is just going to give us a visual to refer to when we're talking about the functions of the nephron, I guess. And where I'm going is all the way back to, I guess that was Unit 1, when we were drawing capillaries. Let's just make this our blood vessel. So if we're thinking about things that are in the bloodstream, in fact, we're about to trace these blood vessels as they move through the kidneys, and we're going to see how some of those tiny capillaries inside the kidney are going to get filtered. Some ions are going to get removed, and other things are going to need to stay in the bloodstream. So my point with this diagram is just to remind us, what are those ions that are found within the blood vessel? I can remember that we talked about those formed elements like red blood cells and white blood cells, and then maybe even those tiny little platelets. But in addition to these formed elements, we said there was the plasma. It was about 55% of the stuff that you would find in the capillaries. These formed elements were about 45%. And we're thinking about the things in the plasma. Most of this stuff in the plasma, I'll just grab a couple of colors here. Remember, most of the stuff was water. It was 90% water. So we can put a lot of these water molecules in here. And this is going to be helpful because when we think about what is in urine, it's going to be leftover things that were in the blood plasma, selective stuff. So there's lots of water in here. I'm not going to be able to draw all of it, but let's just assume lots of water in here. What were those other things that we found in the plasma? Let me get a different color while you're thinking about it. Yes, and that's the color I was looking for. These big plasma proteins made up about 8% of the plasma. I'll just draw these things as these big squiggly lines. I'll just label them as a plasma protein. These plasma proteins are large, so it's going to be difficult for those to squeeze through the... In fact, that might be something that we can remind ourselves too real quick. Since we're doing a nice little review, we've got plasma proteins. I'll keep that review thread that I want to mention in mind in just a second, but just real quick, can you tell me what the other constituent of the plasma was? And I think this was a mix of things, so just throw out whatever you're thinking. Yeah, it was the other solutes, the 2%, and some of those were other ions. Maybe one of the common ions is sodium. We talked a lot about sodium ions. I'm going to be lazy here and just draw potassium ions in the same color. I could even draw those hydrogen ions we talked about. Those we have to keep in close an eye on those, because the free hydrogen ions is what regulates how acidic or how alkaline our bloodstream is. So we have to pay careful attention to those particular ions, because that is pH regulation. There are other ions we might as well mention, some negatively charged ions like this bicarbonate. Let's put a couple of those in there. You'll notice I've got two, just as a side note, we've got 2 bicarbonate molecules and we've got 4 hydrogen ions. So overall, this is somebody's blood that would this be acidic or alkaline blood? It's got more hydrogen ions It is acidic because there are too many of those free hydrogen ions. And can you remind me real quick, if it is acidic, if we're talking about blood that's on the acidic end of the range, just pH scale was 0 to 14, where 7 was kind of right in the middle, 14 is at the high end, 0 is at the low end. At which range would we consider the acidic range of this pH scale? Below 7 or above 7 as our midpoint? That's right. So this is our acidic blood versus basic or alkaline blood. So alkaline is anywhere between 7 and 14. We're going to see that it has, just to remind us, low concentrations of hydrogen ions. Anything that is acidic is going to have more hydrogen ions. I just wanted to mention that because that's sometimes in the introductory biology class that throws people off. Things that are acidic, they have a bunch of hydrogen ions, and that's what gives them a low pH value. The pH value doesn't tell you how many hydrogen ions there. High concentrations of hydrogen ions is a low pH value. Low concentrations of hydrogen ions. In fact, another way of saying low concentrations of hydrogen ions is that there would be more of those bicarbonate ions than there would be hydroxide. Let me try that one more time. Hydroxide ions, I don't know where I'm thinking of that. Hydroxide ion would be one of these things, which we're not seeing. So let's scratch that real quick. We're thinking of alkaline substances that have low concentrations of hydrogen ions, but are going to have a higher concentrations of things like bicarbonate, any of these negatively charged ions, even like that hydroxyl that I erased real quick. All right. So anyway, when I'm thinking about pH regulation, we're thinking about those hydrogen ions. What else in terms of ions? I tried to throw any ion that I could think of. Sodium, potassium. We could have thrown some calcium ions in there. Special attention to the hydrogen ion. What other than ions? And I think that's the only other solute that we've listed so far. Well, nutrients, things like glucose. These little football-shaped things. So remember, if you've eaten a big bag of Halloween candy or something like that, then you've got extra glucose in your bloodstream. This is already, you can see some blood that is becoming very viscous because it's full of a bunch of ions and a bunch of nutrients. If there were, we could include things in here. We saw those high density lipids and low density lipids depending on your diet. We've got glucose, we've got lipids, we've got proteins that can be digested. Like I said, if proteins are digested, one of the waste products is urea. So let's just list a couple of the waste products. So this gives us a nice visual of things that are in the bloodstream. We've got nutrients in there like glucose, things that we would want to keep in the bloodstream. You've got waste like urea that we need to get out. And then we've got these ions that sometimes we might want to hold on to more of those hydrogen ions, other times we might want to get rid of them. So there have to be these cells that can regulate how much of this water do we hold on to, how much of that water do we get rid of, how do we regulate these ions, and how do we get rid of that waste. All right, where's my clicker? Now that we've got a beat on the functions, let's try and zero in to where all of this stuff takes place. It's going to happen inside the kidneys. We have two kidneys. We can see these paired structures on either side of the abdominal cavity. And if we look from above, here they are from above, they're attached to the posterior abdominal wall. We call them retroperitoneal because they lie outside of this peritoneal cavity. I think there's a question on the lecture handout that wants us to see that, first off, these kidneys are retroperitoneal. They lie outside of that peritoneal cavity. They're covered on the outside by this layer of adipose tissue. So that's the perineal fat capsule. I actually don't know if there's a kidney in a bucket back there that we get to cut up. I think there might actually be a kidney in a bucket that we get to cut up today, and I didn't think about that until now. So hopefully that's the case, and we get to see one of these things. And if we get to pull it out of the bucket, it typically still has this perennial fat capsule around it that we'll have to peel away, and then you'll see the surface of the kidney, something that is typically a darker red color on the models that we have. I'll just grab this larger one. We can see this outer capsule. This is the renal capsule. I'm attempting to draw the surface of the kidney in my cross-section sketch. So this outer cover is going to be the fibrous capsule. Let me go back to this slide here. If we go through that fibrous capsule, then we're going to make our way into the kidney. And just to think of this as we're going from the outside into the kidney, kind of the superficial layer of the kidney is going to be this renal cortex. And as we get deeper to the middle of the kidney, we're going to call that the renal medulla. And we know when we've reached the renal medulla because we start to see things like these renal pyramids appear. If I hold up that model that I was just talking about, maybe up here in the light, you can see on here the blood vessels. Maybe I'll just hold it up in front of here. All of these red arteries that are kind of radiating out to the very edge of this kidney. All of this lighter staining area is the renal cortex, and then you can kind of start to see these darker staining pyramid-looking structures that are all kind of huddled in the middle of the kidney. So that's our renal medulla. We're going to try and draw some of those blood vessels on the board. So let's take a step back. What I wanted to show, we've got two kidneys. Each kidney has its own ureter, which is the structure that's going to drain filtrate from the kidneys. And then that filtrate is going to pass through whatever ureter it happens to pass through. Both ureters are going to lead to this urinary bladder. So we have two kidneys, two ureters, but just one urinary bladder. It receives filtrate from both ureters, and then one urethra that drains the urinary bladder. I feel like we've seen that before on these models in lab. I think this was our organ system survey right at the very beginning. We needed to know this as the model of the urinary system. We can see the two kidneys, the two ureters, the one urinary bladder, and then there is a urethra that drains the urinary bladder. This picture just shows us that due to the, looking at this torso model, here's the liver that we talked about. There's this large right side to the liver and a little left side to the liver. It's this large right side to the liver that makes the right kidney sit a little bit lower than the left kidney. So you can see those are not perfectly lined up with one another due to the large right side of the liver. Side note, I don't know why I wanted to mention that, but we need to know the kidneys are outside of that peritoneal cavity. They're retroperitoneal. Here we are finally looking at the surface of the kidney and the structures that I was trying to point out. There's this renal capsule, that outer surface of the kidney. And then once we pierce that, we start to make our way through the kidney. And that outer, kind of this light staining outer section of the kidney is what we call the renal cortex. We're going to see blood vessels that radiate out into that cortex. And then you can see these pyramids, those darker staining areas, the pyramids, as well as you can see this light staining area that I'm pointing to. That light staining area, it's the same color as the cortex out here, but we're going to call those structures the renal columns for whatever reason. Renal pyramids are these triangular structures, and then those light staining areas in between those pyramids, those are renal columns. We're going to see blood vessels in the renal columns and in this renal cortex. And you can at least kind of see the sketch that I'm trying to put on the board here is kind of the base of one of those pyramids. The sharp or the pointy end of the pyramid, this is the apex of the pyramid. At the apex of each of those pyramids, it's not drawn on here yet, but we're going to see those pyramids, those are going to be producing filtrate. So all of the filtrate is going to drain from the apex of these pyramids and enter these calyxes. We're going to call these at the tips of each pyramid. This is a minor calyx. Again, we'll see all of this as we go through the slides. Minor calyxes are like little creeks, and they drain into larger creeks that we call a major calyx. We will define a minor calyx as as something that receives filtrate from just one pyramid. This major calyx right here is a structure that receives filtrate from multiple pyramids. Then we can see, I'm just pointing all of the structures out of the nephron, I guess. This larger opened area inside the middle of the kidney called the renal pelvis, we can describe the renal pelvis as receiving filtrate from all of the major calyxes. So all of the filtrate travels through this renal pelvis, and then from the renal pelvis, it leaves the kidney and travels out of the kidney to the urinary bladder by way of this ureter. But instead of getting into the filtrate tube, let's go all the way back to the start, or just underneath the surface of this kidney, to see how blood flow is going to make its way through the kidney. Again, renal medulla tells us those the pyramids that we were just looking at. So I'm just now remembering that I put these boxes up from the year that I had to give the lectures online, and I wasn't able to point to anything, so I had to use these boxes to describe what I was talking about. So the blue boxes here are pointing to the renal cortex. If we get to structures of the renal medulla, we can really see these renal pyramids. And then I was just talking about the filtrate tubes that gather filtrate either from individual pyramids, there it is, the minor calyx is receiving filtrate from one pyramid. A major calyx is receiving filtrate from multiple pyramids. And then we can see the renal pelvis is receiving filtrate from all of the major calyxes. All of that filtrate that passes through the renal pelvis leaves by way of that ureter, and now we've caught up. And into where my brain was going next, which is the blood flow through the kidney. We'll start with the major blood vessels that take blood into the kidney, and we're going to end with that major blood vessel taking blood out of the kidney. So we're going to go in through this renal artery. We're coming out through the renal vein. I think this is on page 2. So if you are ready, and I'll attempt to draw some of this stuff and label it. That's right, I lost my other marker, so we'll do our best. As we go into the kidneys, we're going to go in through this renal artery. And if we follow, as soon as we get into the kidney, into this renal artery, it's going to split and branch into what we're going to call segmental arteries. It's labeled right here. These segmental arteries are the first branch off of the renal artery. I'm not yet to the point where I can draw those on the board. But if you can see, we go from renal artery to segmental artery. From segmental artery, look how there's some branching in the segmental arteries, and then what those segmental arteries end up leading into are these renal pyramids, these light staining areas in between the pyramids. So in the renal columns there between the pyramids, those blood vessels are going to get a new name. We're going to call those interlobar arteries. In fact, I can start right here. Interlobar arteries are going to be these ones that are going to be along the side of the pyramid, not along the base of the pyramid yet. So let's label this thing as our interlobar artery. Interlobar arteries, once they make this arc and are around the base of this pyramid, we're going to call them arcuate arteries. So let's see if we have them labeled here. Arcuate arteries, you can see, are the arteries, or we'll even call them arcuate veins if we were to draw the veins. They're found along the base of the pyramid. So let's call this arcuate artery. There's our arcuate artery. If we follow this arcuate artery out, we're going to see that as we get into the cortex, there are branches off of the arcuate artery that radiate out into the cortex. There's a bunch of them. You can see all of these little squiggly lines that are radiating out into the cortex. These are called two names. They're either called cortical radiate arteries because, like their name is telling us, they're radiating out into the cortex. Or there's another name, an older name. These arteries are also called interlobular arteries. That sounds very similar to interlobar. So let's put, I sometimes label these things as cortical radiate arteries, but let's just put them both down. This is our I'll label it on this side. Or we can call this thing interlobular. So interlobar and interlobular sound very similar, so if you're going to use that term, be careful. Cortical radiate or interlobular describe these arteries that radiate out into the cortex. We can see a bunch of them. Now here's where we're going to zoom into this cortical radiate artery. This thing that's branching and making its way up, that is our cortical radiate artery or interlobular artery. Here it is. And as we follow it up, we can start to see there are a bunch of branches that branch off of that. There's one, there's one, there's one, there's another one. I'm going to pick one of these and label it. And here we go. Let's just pull this thing out. We're going to see it's going to lead into this tangled bed of capillaries, and then it's going to move away. Hopefully I've given myself enough room to write all of this stuff. We can see branching off of this cortical radiate artery are going to be arteries that we're going to call afferent arterioles. And there's a lot going on in this diagram, but what we can see for the first time are these yellow tubes. That's part of the nephron. So we're going to describe the parts of the nephron and what's going on in each part of the nephron. But right now we're just seeing how this nephron is supplied with blood. We're just drawing the blood vessels yet. And if I can grab a different color, maybe we can add something in here. We're going to see that the nephron is going to start right there around this blood vessel that we're going to call the glomerulus. I'm just going to label this thing maybe nephron. The start of this nephron, we haven't really drawn all of it yet, but we've started it. And we can see that nephron, it's kind of like this Pac-Man structure that is wrapping around this tangled bed of capillaries. So let's label these blood vessels. We know that this one, and maybe another thing that I could add to this that might be helpful is blood is going to be going towards this tangled bed of capillaries through what we're calling the afferent arteriole. Afferent arteriole leads to this tangled capillary bed that we're going to call the glomerulus. So glomerulus is that tangled capillary bed that the nephron is wrapping around. And then we can see blood moving away from this glomerulus by way of what we're going to call the efferent arteriole. Let me see if I can squeeze that in right here. So efferent arteriole takes us away from that glomerulus. Let's go ahead and do this too. I'm going to put a very abbreviated nephron. I'll try. Squiggly little line here. We're going to see there's this long loop of the nephron dropping down. Another squiggly line and a collecting duct. So we've got parts of the nephron. We're going to label the parts of the nephron real quick, but I just wanted to label these blood vessels as we move along the nephron. And I'll abbreviate just parts of this. We will elaborate. We're going to see that the first part of this nephron tube, we're going to call the proximal convoluted tubule. And I'm abbreviating this for right now, just to save myself some space. There's this loop that we're going to call the loop of Henle. We'll see that there's a descending limb to that loop of Henle and an ascending limb. Different things are happening on either side of it. But once we make our way up this ascending limb of the loop of Henle, this last squiggly part we're going to call the distal convoluted tubule, DCT. And then the last tube that I have here is going to be what we call the collecting duct. And if we follow that collecting duct all the way out, that is where urine is going to be produced. That's the filtrate that leaves the nephrons, makes its way into the minor calyx, the major calyx, out the renal pelvis, and eventually through the ureter to the urinary bladder. And so just a quick rundown of the structures to the nephron that we're going to abbreviate. Now that we have those up, I wanted to kind of continue this efferent arteriole because it's all tangled up right here. If you have these diagrams also on the lecture handout, you'll see that if we follow this efferent arteriole, I'm going to back it up just a hair. This efferent arteriole is going to kind of take a little detour, and it's going to actually start to move right along this proximal convoluted tubule, and it's going to move next to this distal convoluted tubule. Back up just a little bit. We're going to see that it then kind of makes a little turn, and it's going to drop right behind that distal convoluted tubule, so I'll draw it kind of popping back out here. It's going to then kind of drop between this loop of Henle and the collecting duct. Here's where I need to change colors a little bit. We're going to see this blood vessel is going to kind of loop around this loop of Henle. And on its way back up, we're going to draw it in a different color. So we're going to see blood vessels as they wrap around parts of the nephron. We're going to label these parts of the blood vessel close to the proximal convoluted tubule and distal convoluted tubule, kind of this part and that part. Those are going to be called paratubular capillaries. With my pointer here, we can point out afferent arteriole. It goes into this glomerulus. Then as we leave this glomerulus, we're leaving by way of these efferent arterioles. And look what they've done with this efferent arteriole. They're showing two branches. I've just drawn one branch on the board, but you can see part of this efferent arteriole that comes around here and wraps around the proximal and distal convoluted tubules. We call that blood vessel the paratubular capillary. And then we can see the branch of this efferent arteriole that dropped down to and kind of wrapped around this loop of Henle. It's like a net of blood vessels that wraps around this loop of Henle. That net of blood vessels we're going to call the vasa recta. And I don't know that that's even labeled on there. So let's call this thing, this blood vessel here. I'm just going to come down here and label it because it would include both sides of this blood vessel. On either side of that loop of Henle, this is the vasa recta. It looks like we're losing juice in this thing. So paratubular capillaries and vasa recta are part of the blood flow through the nephron. If we can look more closely at these nephrons. The one that I'm drawing here, you can see, has a loop of Henle that doesn't quite penetrate very far, if at all, down into this pyramid because I didn't want to overlap how I was labeling things. But when we start to compare nephrons, we're going to see that there are two types of nephrons, what we call cortical nephrons. These cortical nephrons are almost entirely in the cortex of the kidney, and very little of them ever make their way down into this medulla region. Most nephrons are cortical nephrons, and let me say this, depending on what mammal species you are, you have a different number of either cortical nephrons or juxtamedullary nephrons. These juxtamedullary nephrons are the ones that have really long loops of Henle. We will see, hopefully by the end of this lecture, that as we go deeper into the kidney, the extracellular fluid, the fluid in between the cells of the kidney itself, it starts to get more and more salty, the extracellular fluid does, as we get deeper into the kidney. We're going to see that that's actually going to allow these nephrons that drop deeper down into this medulla, they're going to be better at water conservation. Again, this is a side note, but if you were to look at, like if we just walk out in the hall and we found a nephron from a desert mouse, or even like a whale, like these things, or dolphin or something like that, these are mammals like us that, like a little desert mouse or something, it might not be anywhere near available water. So they've adapted to being able to survive just off of licking dewdrops off of plants in the morning. They can survive on very little water. And one of the reasons that those desert mammals can, and the whale is the same way, they get their water out of the fish that they eat. They can't drink that ocean water that they're surrounded by. So they have very well adapted nephrons that are excellent at water conservation. And if you were to look at their nephrons, you'd see a much higher concentration of these juxtamedullary nephrons. I guess that's my long way of saying that mammals that are adapted for desert environments have more of these juxtamedullary nephrons. We are not adapted for desert survival. We have more of these cortical nephrons, and sure enough, I don't have to walk 25 yards before I find a water fountain. So we are used to having water close by. We can tell that just by looking at our nephrons. Let me pull away from nephron comparison real quick to get back to blood flow through the kidneys. From the diagram over here, I was labeling everything until we got to this dotted line here. The dotted lines just represent the parts of the blood vessel that pass through the nephron. So I tried to sketch a quick nephron. We can see afferent arterioles are what deliver blood to that glomerulus. Then carrying blood away from the glomerulus is the efferent arteriole, and then depending on where that efferent arteriole goes, if it stays around the proximal or distal convoluted tubule, we call it the paratubular capillaries, and like we said, if it drops down around this loop of Henle, we're going to call it the vasa recta. Then what I was about to get to is if we were to continue to follow this vasa recta, we would see that all of this now blood that's going to be flowing through the venule side of a capillary are going to get picked up by what we call cortical radiate veins. Just like this artery is radiating out into the cortex, we would have seen this vein too radiate out into the cortex. I don't want to write over what I've labeled here, so we just have to imagine this ascending blood vessel here. That's going to end up making its way into these cortical radiate veins. If we go back to this picture, there are those cortical radiate veins kind of running parallel to the cortical radiate arteries. If we go back to that cortical radiate vein, cortical radiate veins are actually going to end up merging into arcuate veins. So running parallel to the arcuate vein, let me say that again, running parallel to the arcuate artery, there are going to be these arcuate veins, and then interlobar veins. I'll just let the color designate that without writing more on that. Getting to this picture, we can see interlobar arteries. They're these squiggly little lines that radiate out into the cortex, and some of those are going to be delivering their blood down there into those arcuate veins. So I've just drawn an arcuate vein along the base of the pyramid. If we follow that arcuate vein, it's going to drop down to the side of the pyramid into what we would call the interlobar vein. The blue one is the interlobar vein. From interlobar veins, here's a difference between the arteries and the veins. In the vein side, these interlobar veins run directly into the renal vein. There are no segmental veins where there were segmental arteries. So hopefully that got us all the way through the flow chart. If I go back to this picture, that points out the vein side, how the vein side is different from the artery side in the sense that the artery side has the segmental arteries in between the renal artery and the interlobar artery. That's not the case on the vein side. It goes directly from interlobar to renal vein, and then from renal vein right into that inferior vena cava. The major blood vessels that we need to remember are going to be these ones that are around the nephron, and then anything that we can find on the lab models. So our large kidney here, we can see our renal artery that goes in there. We can see segmental arteries. Then there's the interlobar artery, arcuate arteries, and cortical radiate arteries. If we want to get any closer than that, we have to zoom in. Just grab these. Now we've got one other model of a kidney that zooms in to just one individual pyramid. I'll just try and hold this up. From looking at one individual pyramid, kind of like I was drawing over here, along the side of this we have an interlobar artery and interlobar vein. When those make their way along the base of this pyramid, they're segmental arteries and veins, and we can see radiating out into this cortex, there's a cortical radiate artery and cortical radiate vein. The white tubes there are the nephrons, so we'll use these models to point out parts of the nephron and see paratubular capillaries or vasa recta. Okay, so we've made it through arteries and veins. Any questions about that blood flow through the kidney? Did we get all of that in one quick swoop? Okay, good. Then with With that out of the way, what I want to do then is draw parts of the nephron. And I'm thinking if we can at least remember this cortical radiate artery, the afferent arteriole, glomerulus, and efferent arteriole, I can erase this stuff. And we're going to zoom into just this part of the graph and stretch our nephron out a little bit bigger so that we can see where filtration happens, where reabsorption happens, where secretion happens on different parts of this nephron. And looking at our picture, we can see this is supposed to represent the base of one of these pyramids. We've got our interlobar arteries and veins. What? Oh, sorry. This is even more zoomed in than that. Luckily, I have the label here. If this is an interlobular artery and vein, then we're talking about the same thing as our cortical radiate artery or vein. Branching off of that cortical radiate artery or vein would be this afferent arteriole. So they've labeled that as an A. We know that that's carrying blood into this glomerulus. Now, what's interesting about this picture, you can see just like the model that we have, I was about to hold up, this is, we can see this glomerulus, this tangled bed of capillaries inside this nephron model. And what they've done is they've basically revealed this glomerulus on half of this nephron model. And then the other half, you can see it's covered up by these cells that we're going to call podocytes. We're going to see that they make up the visceral layer of what we're going to call the glomerular capsule or Bowman's capsule. The glomerulus is surrounded by a capsule of epithelial cells. We'll look at them more closely in a second, but just quickly going with the blood flow, we know this glomerulus, if we were a red blood cell moving through the glomerulus, we would eventually leave that glomerulus by way of the efferent arteriole and end up in the peritubular capillaries surrounding that proximal or distal convoluted tubule, or maybe we would surround this loop of Henle, in which case we would be the vasa recta. This is showing how the branches of the veins that come off of this vasa recta make their way down into this interlobular vein or cortical radiate vein that goes down and hooks up with that arcuate vein and interlobar vein. So back to this picture of the nephron. We're going to focus in on the nephron itself. And I guess that's the last slide of part one. It's actually the first slide of part two. In part two of this PowerPoint, we look at all of this stuff. We look at the nephron and we talk about what happens at each segment of the nephron. So let me make myself a little bit of space. We basically spend the rest of the time talking about this slide, even though we've got multiple pictures of the same thing. I'm just going to start by trying to draw part of this nephron, and we'll draw the blood vessels around it. Okay, so we've got our nephron sketch. Parts of the nephron are going to include this first structure that's called Bowman's capsule. It's also called the glomerular capsule. When we look more closely, we're going to see Bowman's capsule has two sides. It has a parietal layer and a visceral layer. The visceral layer are those podocytes. So we'll look closely at what's going on here because this is really the vacuum where filtration is occurring. We know that, I'll just draw one of our cortical radiate arteries, and branching off of that cortical radiate artery is an afferent arteriole delivering blood to this glomerulus. And then we have an efferent arteriole that goes away and then would eventually kind of start to follow this proximal convoluted tubule. Let's do that. Let's just go ahead and squiggle this thing in here. Squiggle it down here. I'll just leave myself some room to label things. Okay, so we've got a little reminder of the blood flow. If we want to list this, cortical radiate artery gives way to our afferent arteriole. You know, the diagram you have in your class handout is even different than this, so maybe it's helpful to label this a couple of different ways so that you can more easily label the one that you have. Right there in the middle is the glomerulus. And then away from the glomerulus, we have our efferent arteriole. There's our efferent arteriole. If we continue to go down that efferent arteriole, this part that surrounds the distal convoluted tubule and our proximal convoluted tubule are the peritubular capillaries. And then eventually down here we have our vasa recta. Excellent. Now we can talk about the parts of the nephron and what's happening during each part of the nephron. This Bowman's capsule is going to be where filtration occurs. And what I mean by that is, and again we see this in slides, so I'm not sure why I'm wanting to draw everything by hand, but if we go back here to what is our representation of a capillary, remember that the capillary is made out of single layers of these squamous shaped cells. So when we were drawing these things before, we can add this simple squamous shape to our little representation of this capillary wall. It's a single layer of these squamous shaped cells. So just to remind us, and I need more colors here. Hopefully that's enough. Any time that blood pressure is so high or things are small enough like water or any of these ions, those things are small enough that they can squeeze right between those spaces, between these cells of the capillary. This is what we call filtration. And it happens in the glomerulus, one of the main places where this filtration occurs. So filtration are things squeezing between the cells of the capillary, and if you're small enough to fit through those gaps, then filtration is going to occur. Turns out some of the things that are small enough to squeeze through those gaps we actually don't want to lose. So glucose is one of these things that can squeeze through that gap. And if glucose left the blood vessel and was going to end up in the urine, that would be bad for the cells that could use it as a fuel source. So we're going to see that when things are pulled back across and pulled back into the blood vessel, let me get this out of the way, we're going to describe this as reabsorption because it was already in the bloodstream. It left the bloodstream for just a little bit and then was pulled back in. So reabsorption. When we were talking about the digestive system last time, we said in the small intestines is where absorption occurred. That's the first time we picked up this glucose molecule or picked up this water. We ingested it. It got absorbed by the small intestines, and that's how it got in the bloodstream. If it leaves the bloodstream, we've lost it. It could end up in the urine unless it's reclaimed or reabsorbed. And I guess I'll just mention the last thing that we'll see. Sometimes waste products like urea are going to be secreted out. So just a comparison between this term filtration, which is just particles making their way out of the capillaries because they can squeeze through those little gaps. Not everything that is filtered is going to be lost in the urine. Some of it is going to be reclaimed through reabsorption. And then we'll even see that some parts of the nephron are going to be capable of secretion That happens in this distal convoluted tubule and the collecting duct. We said some of those cells, as we will draw in a second, some of the cells of the distal convoluted tubule that line that distal convoluted tubule and line this collecting duct are capable of ion regulation. That's why they have these arrows with dotted lines. It just indicates in this diagram that this segment of the nephron can change how it operates depending on certain hormonal cues. So we know there are hormones that we mentioned that can affect the kidney and cause the kidney to hold on to more water. That was our antidiuretic hormone. We know that there's hormones that can affect the kidney that get it to hold on to more salt, more sodium. That was our aldosterone. We'll see that aldosterone and antidiuretic hormone really work in the distal convoluted tubule and collecting duct. There they can regulate, do we need to hold on to more hydrogen ions? Or if we have too many of them, maybe we need to secrete and get rid of some of these hydrogen ions. So just know the difference between filtration, reabsorption, and secretion. As we go back to this diagram, filtration happens right here in the glomerulus. So if you're small enough to make your way through those little pores, and if you remember when we were talking about capillaries, we said there's 3 types of capillaries. The one that I'm referring to here are these continuous capillaries whose cells don't have any holes in them. But if you look closely at this particular capillary here that we call the glomerulus, you'll see it has a bunch of those tiny pores punched in it. We call this fenestrated capillaries. And those tiny little pores, there's a better picture of this in a second. The tiny little pores that are all along this glomerulus make it easy for filtration to occur. But like we said, that means anything that's small enough to make its way into these tubes is going to make its way out. So things like sodium ions, I'm kind of wishing I would have given myself a little bit more room to write here, but sodium ions, potassium ions, things like hydrogen ions. Well, let's put some other things in there like water. Water is small enough to fit through. Nutrients like glucose is small enough to fit through. So I'll put my little hexagon shapes that represent glucose. Anything that's small enough to make its way through those little pores is going to make its way into the first part of the nephron, which I'm realizing we haven't labeled on here yet. So this part, from here to here, is going to be our proximal convoluted tubule. Proximal convoluted tubule gives, if we ride through this proximal convoluted tubule, at the end of it, it makes a deep dive down what we're going to call the loop of Henle. To be clear, this descending limb of the loop of Henle comes first, so we'll label that descending limb. We're going to see this filtrate, any of the stuff that made its way into this proximal convoluted tubule that doesn't get pulled out of the proximal convoluted tubule is going to make its way down this loop of Henle. We'll talk about the different sides of the loop of Henle. We've got a descending limb. Then up here we have an ascending limb. Anything on this side is the ascending limb. So there's the first part of it that is considered the thin segment of this ascending limb, and then there's the thick segment. I'll say what's happening in both of those in just a second. Let me label the last part, which we can consider the distal convoluted tubule. This distal convoluted tubule, if we ride through that, we end up in this collecting duct. So I'll just label this thing as a collecting duct. What I want to point out here is this collecting duct. Collecting ducts are shared by multiple nephrons. So you can see what we've kind of drawn a distal convoluted tubule taking its filtrate into this collecting duct. And the other thing that I was drawing in here, I can even add another one up here, this would be a distal convoluted tubule from a totally other nephron that we didn't draw on here, dropping its filtrate into this collecting duct. So multiple distal convoluted tubules are all sharing one collecting duct. This collecting duct, after receiving filtrate from all of those distal convoluted tubules, is going to be dropping that. What comes out of the collecting duct is urine. It travels through those calyxes, through the renal pelvis, through the ureters into that urinary bladder. So let's back up a second. Let's go back to these parts of the nephron. Now that we got it labeled, again, this is already on the board, but let's just talk our way through this. What happens in the proximal convoluted tubule is that this part of the nephron is going to try and reclaim anything that can be reused. For example, one of the things that we have there are sodium ions, there's potassium ions, there's glucose. That can be reused, so we wouldn't want to waste glucose or certain ions in the urine. I guess we could also go down here and say something about what ends up in the urine to not give this away. Urine is going to be mostly things like water, urea, and ions. Primarily lots of sodium ions, maybe things like potassium ions or one of these bicarbonate ions. Even though, you know, we're going to pull a lot of stuff out of the urine's still going to have a lot of water, a lot of sodium, and some waste in it. So not everything is going to get reclaimed, but when we look at the proximal convoluted tubule, it's going to do its best to pull any of the useful stuff out of the filtrate so that it doesn't end up down there in the urine. And you can see it's able to reclaim 100% of these glucose molecules. So the proximal convoluted tubule, we're going to see it's going to reabsorb the glucose molecules. It pulls it out of the proximal convoluted tubule, and those glucose molecules are going to go right back into the bloodstream.